I have started the process. In a couple of minutes, we'll be live.
So hello, everyone. I understand folks are logging in for the upcoming session. We'll get started at the top of the hour. If you have questions for Dr. Melanie Ott, today's guest, you'll notice there's a chat icon at the bottom of your screen. Please use that, and I'll try to get as many questions as possible.
We've got about a minute or two, and we are successfully streaming, so always good to go.
And you can hear me okay, right?
Yes.
Great.
So are we on?
Almost. I'm gonna start in just about 1 minute. Give folks a little bit more time to get settled.
Okay, perfect. And we're starting with a question about where we are with the science?
Yes, but I'm gonna talk to you about the link that I sent you today too. We'll start with that. Okay, let's get going. Hi, everyone, and welcome to our third webinar focused on the COVID-19 pandemic. I'm Chris Rebelo. I'm the Director of Communications here at the Buck Institute for Research on Aging, and I'll be moderating today's conversation with Dr. Melanie Ott, a senior investigator at the Gladstone Institute for Virology and Immunology and an adjunct faculty member here at the Buck. Melanie is a virologist. She stares down this coronavirus responsible for COVID-19 every day. She is part of an international collaboration that is looking at existing drugs that show some promise against the disease. She's also developing a test that could be widely used to screen for those who are infected with the coronavirus. So today, we get an important evidence-based perspective on the science of COVID-19. As ever, we welcome you to be an active participant in this session. You should see a chat widget at the bottom right of your screen. Please use that feature if you'd like to pose a question for Dr. Ott. I'll try to get to as many of them as we can. So Melanie, I know you're busy. Thanks for making the time to be with us today. The timing is perfect. I want to first ask you a question about some, quote, breaking news that came out this morning. Gilead Science, which is a Bay Area company, announced that they are aware of positive data from a study of its drug, I hope I pronounce it right, remdesivir. They say it shows promise. There's been no comment from the government yet. I know there's some controversy about the drug, but overall, can you talk about what is this drug and what does it do?
Yes, good morning, Chris. Thanks so much for having me. It's indeed an exciting and, as you can imagine, very busy time for virologists and for the entire Gladstone Virology Institute. Yes, remdesivir is a drug that many have watched very closely to see how it does against the virus. And I think the news today from Gilead itself appears promising. That in their clinical trial that they see efficacy here against COVID-19. I understand that there is a second trial from the NIAID where we still need results from. This is actually the more important trial in the way that it actually compares the drug between untreated and non-treated patients, and I'm really eager to see the results. but at least there is some hope here, and we're certainly excited to see the full extent of the clinical data.
And this drug is an antiviral, right? So would it block replication?
Yes, I think this is a drug that was initially developed against Ebola. And it targets an enzyme in the SARS-CoV-2 virus that's called the polymerase. This enzyme is actually amplifying viral genomes during the course of the infection, and basically allows the virus to amplify and to spread. Now, this is a very important step, obviously, and the idea and the evidence currently that we have is that that drug from Gilead directly interferes with this amplification.
Great, thank you. Well, we'll all keep track of the news as it unfolds on this one. So let's take a step back. You're a virologist. Where are we now in the science and in the research? Are we getting close? Are we at the, where are we?
I think we are very much at the beginning. I think the time that we are in very much reminds me of the beginning of the AIDS epidemic, some 30 years ago. I think like then, we're dealing with a completely new and deadly virus that jumped from animals to humans, and we're having to learn everything about it. So the point is that we are very similar stage, very much at the beginning, but I think the big difference from the beginning of the AIDS epidemic and now is that we are doing science really in a different way today. We are learning much faster, and we are sharing this knowledge much faster. And we're also doing science in a different way today. I think back then we had to go and work on each of the individual proteins one by one. Now we can really take a more holistic approach and look at everything at the same time and generate a lot more data, and because of the existing technology. So I think it's as I said, a very beginning time. But I think it's also a very, exciting times because we can accelerate so much faster.
That's great. Is there anything about this coronavirus that is, quote, special, or is there, like, where's our biggest gap as far as understanding about the virus or what happens in our bodies?
Yes, so the bottom line is that we have to understand everything about this coronavirus. There's nothing specific about that virus that makes it different from other viruses. It's a unique virus, but like any other virus, we have to understand how it's been transmitted, how it replicates in the cells, and how it affects our body. How it makes us sick. And I think, all this knowledge ultimately feeds into better testing, better treatments, and ultimately, hopefully soon, a vaccine.
That's great. So one of the biggest debates in this country and around the world is it time to open up? We need to open up. The economies are suffering. There are some states that have opened up a lot more than here in California. What's your reaction to that?
Well, this is risky business. I think it's risky in the way that we cannot open up and repeat all the mistakes that we have done a few weeks back when we started sheltering in place. I think we want to wind back time, but we want to do it right this time. And I think what we need is really, to focus on three questions. We really need to focus on testing. And we need to, identify people who are infected. We need to have reliable and frequent testing so that we can do, contact tracing, and we can isolate people who are being infected. This is, I think, a cornerstone for opening up and for going back to a new normal, basically.
So I got a question from somebody: how wide must testing be? For example, 50% of the population, on an ongoing basis if carriers are asymptomatic. So talk about the the degree of testing that you think is going to be needed to really get a handle on this?
I think we have to absolutely increase our current testing capacity. I think the governor has talked about some numbers that he thinks we should reach in the 60,000s. And I think these are probably good guiding numbers. I think currently the point is that we are not there. And that we have to increase this capacity to whatever we can do. I think I see not only a bottleneck currently in the testing availability, I also see a bottleneck in the way how we prescribe testing. I think many people who would like to get tested cannot get tested. But at the same time now, universities and blood banks are rolling out more systematic testing across the country to really do community-based testing independently of symptoms. And I think this is an important second arm to what we can do to really get on top of this, on top of this virus.
So there's also emerging testing for antibodies to see who has had this virus and who may have immunity. So can you talk, I know you're a virologist, based on what you know now, do you think that there will be immunity to this? And what do you think of antibody testing and how useful that may or may not be?
Yes, so I'm currently working at Gladstone on a test that is directly testing for the virus, combined with actually contact tracing, and I think that's one important arm currently. And as you mentioned, the second important arm is that we not only look at the acute infection to see who is carrying the virus and shedding the virus currently, but we're also looking into who has had the virus in the past and who might be immune. And you might notice that I say might, because we don't really know whether the current antibody testing is really translating into immunity. And this is an open question that we need to answer pretty fast. I think the other open question currently with the serology testing is that they have been rushed on the market very fast. And I think we have to take a step back currently and really make sure that they have all the appropriate quality controls in place so that we can trust the results.
So one of the questions I was going to ask you later is, do we have the test yet? So it seems like we don't have the test that everybody can go, this is reliable.
I think there will be no the test, because I think there's different tests for different things. As I said, we can test acute patients, mostly with nasal swabs, for the existence of the virus in these swabs. And we can test in the blood patients who might have, or people who might have had the virus. So these are totally different approaches. They take different samples, they take different technology. And so they will have to be combined in the future to really get the full handle on the virus.
So let's talk about the vaccine. There's been something in the press with a group out of Oxford that seems to be ahead of the game, and there's some talk about maybe a vaccine being available this fall. When you read the literature, where do you think we are with vaccine development?
Well, I think we are actually at an enormous pace currently with the vaccine development compared to other vaccines that we have developed in the past that takes years to do. And I think the reason why we are moving so fast, and most of the companies that are claiming, early victory here are those that are going after the RNA of the virus. So this is, since the virus genome sequence was deposited in early January by our colleagues in China, I think this genome has basically provided the basis for most of the research that's currently ongoing and around the world. You can use it to develop diagnostics, you can use it to develop therapeutics, and you can use it to develop a vaccine. And so what has happened is that genomic sequence, which is just the information, the genomic information of the virus, was used to directly translate it into a vaccine. Usually what we do is we might generate, parts of the virus and then produce them in a very specific way and then inject them to generate immunity. But here, basically, this was bypassed, and we take the genomic information, inject it, and then let the body do all the work. This is a wonderful concept. And it was extremely fast, and that's why people talking about maybe having something early on. But at this point, we don't know if it works because it has never worked before. It has never been used before. And so we don't know if it's going to work as we predict it will work.
Great. So I just got a really good question about viral load. And I think it's been in the press that healthcare workers who are obviously exposed to this virus in a large degree are getting sick and sicker. Somebody wants to know, do you know the minimum amount of virus? Like I've asked myself this question. If I'm out for a walk and there's somebody maybe 4 feet away who sheds a little virus, does it only just take a little virus to get an infection? How does that factor in?
This is a very good question, one that we're dealing with a lot because viral titers is what my daily life is all about. And so the answer is we don't know how much virus you need to get infected. But what we know is that, or what we think we know is that the amount of the virus that you get in contact with might determine how sick you get. And so this is a good example of the healthcare workers that in general are getting more sick, and although they're young and healthy and should not have any coexisting conditions or risk factors in that way. And so one explanation is that they might get into touch with the virus with higher loads of the virus. They might also inhale them more because they are in closer contact with patients. So they get deeper into the lung and can establish infection there. So I would say the question, I think that the amount of virus that you get in contact with might determine how severe of an infection you get, and also potentially where this infection is going to set shop in a body.
Got it. Great. So let's talk about some of the work that you're doing at Gladstone. You are part of an international collaboration of researchers who are looking at some 69 drugs that have been shown to have some promise against COVID-19. What stage of that work are you on, and how are you handling it?
Yes, I think this is a really exciting collaboration that we have with my colleague Nevin Krogan at the Quantitative Biology Institute at UCSF, and also part of Gladstone, and also an adjunct member at the BARC. And we have been actually working together for 2 years now in a very close and coordinated fashion, because our goal was to find host-targeted therapies. And this is different from what I said before about the Gilead drug, which is really targeting a viral enzyme. We were interested to really look in the interaction between the virus and the host. And here it's really important to remember that a virus cannot exist in a vacuum. It is very dependent on finding a host cell that it can hijack and then turn into little viral production factories. And so Nevin and we have started a joint program called BioFulcrum, where we look at multiple viruses and their host interactions. And it is really a combination of the expertise of our 2 labs, Nevin being really an expert in something called mass spectrometry, where you can look at the proteins and identify all of the host proteins that are making contact with the virus in the cell. And my lab has focused on generating advanced technology to actually study viruses and cells. So we create little mini organs in a dish, and then infect these mini organs called organoids to find a better way to really predict how these viruses are going to act in the real organ and in a patient. And so by combining these 2 things, him identifying the factors and us testing whether the factors are important and what role the factors are playing in the infection, we have established a whole platform where we're looking at multiple viruses, comparing their dependencies on the host cells, and actually using these shared dependencies as targets for new therapeutics.
So I understand that as far as the work on COVID-19, you've actually created little like mini lungs, which seems like it's really an important thing to do because this coronavirus does really impact the lungs, right?
Yes, absolutely. So I think the reason why my lab became so early interested in studying SARS-CoV-2 was that we had certain tools in the lab that I thought were really relevant for studying this virus. One was this collaboration with Nevin and this platform that we had established. But second was that we had within this collaboration just started to study influenza virus in lung organoids. And this was very promising, and we have established together also with Jennifer Doudna's lab and her CRISPR technology, ways how we can inactivate these individual mechanisms in the cells that throw a lifeline for the virus. And so we thought that we could use this knowledge and these lung organoids and this technology to immediately translate it to SARS-CoV-2, and that's what we're doing currently.
So I got a question previously from somebody who's really curious, what's it take to set up a lab where there's like live viruses happening? What's that involve?
Well, that involves a lot. Thank you for that question, because that's what keeps me up day and night, or has kept me up day and night for the last 2 months. It is not easy to establish a lab like this. You need a very high biocontainment level. Everything is regulated. You enter the laboratory, the laboratory has to be built in a certain way so that there is no air coming out of the lab into the outside. And also everything that is in the lab cannot leave that lab unless it goes through an autoclave, which is basically a high temperature oven that kills everything when it goes through this. So there's certain logistics and facility requirements. But there's also a lot of paperwork that is involved to establish standard operating procedures that will be strictly followed so that there's absolutely no risk for anybody in that laboratory to get infected. And I think the basics is that we work with respirators and whole body suits. It really looks a little bit like a moon landing when you are in there. But since it is a respiratory virus, you really cannot be around the virus and not be protected in a very systematic way. Now, a lot of people think, is it really safe to work in there? And I keep saying that I'm convinced that it's currently more safe to work in this laboratory with all the protections and precautions that we take than it is to go actually to the grocery store around the corner currently.
So when you go, so when you go to the grocery store, what do?
I put my mask on and I wash my hands after.
That's great. So the work that, we talked with Larry Brilliant about this the other day briefly, and there was an article in the New York Times this weekend that talked about the future of pandemics and that we're probably not done with them for sure. And that there are efforts to develop drugs and vaccines that are pan-antivirals. So it sounds like maybe some of the work that you're doing inside the cell, first of all, what do you think of pan treatments? And is your work, could that contribute to that?
Yes, we hope so. I think that's the goal. I think that's why we started the BioFulcrum effort 2 years ago. With the dedicated, goal to develop pan-antivirals. And the reason why we thought the host is a good target for this is because we have learned through our ongoing studies with Nevin and in my lab that there is actually a lot of overlap of these factors in the cell that are important for viruses. Although viruses are very different, they look different, they occupy different spaces in the cell, they still need energy to replicate. They need, they need certain nucleic acids to build more genomes. So there's a lot of bottom line that we find is actually common between these viruses, and they have to deal with a lot of immunity and restriction factors in the cells. And so I think our approach was to take advantage of these shared pathways that we target, and so then with the hope that a future virus, about which we don't know yet at that time much at the beginning, would also follow the same principle and require these factors, and it would respond to these drugs. So I think we're 100% aligned with this article that you mentioned. I think the remdesivir study is also a pan-antiviral in some sense, because it was developed against another virus and is currently being used, against the SARS-CoV-2. And we have other examples. I think HIV drugs have been used against, COVID-19. And so there's a lot of, efforts currently ongoing to see what we can borrow from other viruses and can translate it now to this current virus.
Great. So I got a question. Viral immunity is mainly related to cellular immune responses by T cells and other adaptive immune responses. Where is the research on T cells? Antibodies are fine, but limited in their effects. So can you give us a little tutorial about T cells?
Yes, absolutely. I think my research is mainly focused on the infected cell, and that's the innate immune response, but as the question correctly points out. But eventually these innate responses call in the adaptive arm of the immunity, which is our dedicated immune cells, T cells, B cells, macrophages, and others. And I think T cells are a very important part of this because they provide important help for the B cells to actually generate the antibodies, and they also directly destroy the infected cell when they recognize it. And the adaptive immunity is really something that can go very specifically after the infected cell. So that's something that a vaccine is trying to harness and to improve. We know we start to understand more about T cells, especially also in the severe disease, because we know that the immune system is likely responsible in part for causing this severe lung disease. So there's a lot of effort currently been focused on understanding what's happening in the lung and what might tip the immune system to an overreaction versus an appropriate reaction. And I think one thing is that we're learning these things now, maybe at a slower pace than we wish, but we need access to T cells and blood samples from patients currently in the clinic. And there has been huge studies being set up for this. For this, you need appropriate institutional permits and study protocols. You cannot just take something and study it. You have to. This is highly regulated. These have been set up. Now the good news is in California we have not enough patients that we can enroll in this because we're doing extremely well in flattening the curve, and in New York, people are overwhelmed to actually do systematic studies currently because of the need. So I think this will balance out in the long run, but I think we will learn a lot more about the immune response and the T cell in the coming weeks.
So as you talk about innate immunity, but then you get into T cells, I'm starting to ask myself, I'm sure other people are, well, is there anything I can do to make my T cells happy so that if I get infected, they'll be ready? Or what can I do to get my innate immune system geared up so in case a piece of that virus lands on my head, I'll have a chance of either not responding to it or having a very, very mild response. So what can people do?
I think you can stay healthy. You can nurture yourself and make sure that you are in the best shape ever. But I think most importantly, to prevent infection, you have to follow the guidelines. You have to wash your hands, you have to wear a mask in public, and you have to keep your distance so that your T cells are as little engaged as possible.
And you think that we're going to be doing the hygiene and social distancing and masks for a while?
I think it will be part of our reopening strategy and maybe also of our new normal, as long as we don't have a vaccine and potentially also not really effective therapeutics. So I expect to have these guidelines around for a while.
Okay, so I want to talk about, get back to the biology and the physiology of COVID-19 and what goes on in the cells. So it's complicated, right? At least from I've read that the virus hijacks proteins, and then proteins start replicating. But there's a lot of connections between all these proteins. Am I correct in that there's a lot to understand here?
Oh, yes, there's an amazing amount of complexity here. You know, that it really shows us that a virus is really a master manipulator in the cell and in the body. And I think we have to understand it on a cellular level, but also on an organismal level, where we really understand how, you know, in the cellular level, we know what the virus does to a cell when the cell enters, but we can also learn what it does to the neighboring cells that might get all its guards up and prevent infection. So we currently really doing these infections in the cell at a single level, where we can sequence the genomes or the genes that are being expressed in these cells on a really whole genome scale. So we know all of the factors that are going up and all of the factors that are going down in a cell when it becomes infected, but also in a cell that sits next to the infected cell and does not become infected. And that cell is almost more important for us to study, because we want to know what effectively is actually preventing the infection in this cell. So there's an enormous amount of complexity already going on in a relatively simple dish that I study in the laboratory. But when you then go into a mouse or into a patient, I think then you see how that cellular level is interacting with the surrounding, and what compensatory mechanisms might be at play to either fight the virus or to help the virus.
So there have been some controversy about various drugs that either show promise or don't. I'm thinking specifically of ACE inhibitors that people take for high blood pressure. Does the complexity of what's going on inside the cell, does that sort of explain some of the up and down responses from some of these drugs?
Yes, I think it really is a perfect illustration that viruses and medicines affect the whole body, and that there is a complex interaction, and that we have to look at the whole thing instead of just you know, at singular aspects of it. And with ACE2, it's really interesting because ACE2 is the receptor of the virus. It really allows the virus to enter the body and to enter the cell and to set shop and produce viral factories. But at the same time, we also know that in the lung, it is an important anti-inflammatory protein, and is important actually for lung health. So we also know that if you take ACE inhibitors, you have a compensatory upregulation of that receptor in the lung. So I think the controversy currently is that you want to, you know, inhibit the virus to enter the cells, and maybe these inhibitors could play a role there. But you also inhibit basically an important lung pathway that could help you deal with the infection better. And then on top of it, the drug might upregulate this, which is good because it contributes to lung health, but it might be bad because it enhances the entry of the virus. And that's the big concern. However, so far the studies, this is just a theoretical concern. I mean, some studies have started to look at, you know, seeing whether patients on ACE2 inhibitors or ACE inhibitors are doing better or worse in the clinic. And there is, to my knowledge, not a conclusive study published currently. So I would say that it's very important before stopping any medication because you become concerned or panicked. I think the important thing is really to work with your doctor, discuss this with your doctor, and if there's any indication for a switch, do this very slowly because you want to avoid getting into uncontrollable hypertension because of the withdrawal of the drug. So I think currently the evidence is there, especially is on the radar, and people are looking into it. The data so far doesn't show a huge difference between people who are taking ACE inhibitors or not, but this has not been systematically studied.
Got it. So there's also been some scary stories about young, healthy people who get infected with COVID-19, and then they have really big strokes. What's going on there?
Yes, I think there is absolutely an emerging picture of the virus interfering or interacting with the blood clotting system in a very specific manner. This is not totally new, but other viruses are doing it too. But the question is this at an unprecedented level? And is this really something that the virus is doing uniquely, or is it just a byproduct of the inflammation that is ongoing? So the blood clotting system is currently under, I would say, intense scrutiny. And we really have to find out what exactly is going on there. Again, this is a very complex system that you do not want to mess with unless you know exactly what you're doing. And the key for us is really to study this better and to see what we can do about it. I understand that doctors in New York are thinking that maybe some of the severe disease that we're seeing in patients is also connected to some infarction in the lung, and are playing around with levels of heparin, which is an anti-clotting medicine, to see whether that improves actually the breathing problem that some people have. So that is definitely an area of intense investigation, not only in the central nervous system, but also in the lung, or even in I think we're seeing even some vascular symptoms in extremities, in legs, or in feet.
Yikes. So I got a question. Does this virus mutate very quickly? So talk about mutations and how that might impact the future.
Yes, I think mutations is a very important part of any virus, because a virus always has a propensity to introduce little arrows into its genome through the polymerase that I mentioned very much at the beginning. And so these random mutations can become very important in the long run when there's a selective pressure. So for example, when I say viruses can develop resistance against drugs relatively fast, especially if it's a drug that targets a particular factor in the virus directly, because then these random mutations occur, and those that refer resistance to a drug will then grow out and become the dominant viral strain. So far for this virus, I think the common assumption is that it does not have a very high mutagenic rate. So the error rate of its polymerase is not as high as other error rates from other viruses. But it has very unique features where it can actually use recombination and grabbing genetic material from other viruses or from other genetic sources to incorporate into its genome. So it's a very, very interesting virus. We have so far, when we look at the sequences of the virus across the world, and there's central databases where all the sequences are being deposited, not seen a huge diversity. We know that there's certain hotspots in the genome where things are happening more rapidly, things are deleted or changes occur in the genetic code. But so far it has not shown sort of a very not a very high mutagenic rate, and it hasn't shown a certain direction and where it's going. But this might change, as I said, if we put some selection pressure onto this virus in the form of growing immunity in the population, a vaccine, or potential therapeutics that might cause emergence of mutant strains.
But for right now, the fact that it seems so stable, that's a good thing, right?
It's a very good news. It's a very good news also for the development of a vaccine, because a virus that has very high mutagenic rates and variability in its sequences are much harder to capture in a vaccine, or also in our own body to develop, really effective neutralizing immunity against it.
So I'm going to snap back to a bigger picture now, because I got a question. Somebody wants to know if you have any comment on the president's cut of NIH funds to the Eco Health Alliance. They look at, there was funding in China to study bats and to look at, to try to find out how viruses jump from bats to eventually us. That funding was cut. What do you think?
Well, I have a lot to say about this in general. I probably keep it short here. I think the point is that if we want a vaccine fast and we want medication fast, we have to fund science. And I think this is not what seems to be happening in this administration here. This principle is not being understood very well. I think this specific effect on this, cutting the research in China, I think is politically, informed or motivated. I think this is a very, very valid research line, because it really helps us understand what is happening in the bat, which is the origin of the coronavirus, the current coronavirus, and former coronaviruses, and in general is a hotbed for viruses. So this work has led to our understanding that there is hundreds or thousands of coronaviruses in bats that we don't understand. It has also informed us to show that this coronavirus has a very close relationship to an existing bat coronavirus that has been sequenced previously, but only because we had scientists working on this. And I think going to the root cause of these viruses, which is the animal host, and finding the intermediate host for these viruses is absolutely important for us to be better prepared for the future, and to actually also cut, the current pandemic.
Because as we talked earlier, there's more coming, right?
Well, I think if you look at the current frequencies of outbreaks that we have every probably 5 years currently, I mean, starting with SARS, going to MERS, looking at the Ebola outbreaks, looking at Zika a few years ago, now SARS-CoV-2, I think we can anticipate that this rhythm is going to continue and potentially accelerate. And I think this is because we have a much more connected world, and a much more crowded world also in terms of transmission. And I think we will have to deal with these outbreaks in the future, and hopefully we will be better prepared. And cutting funding for initiatives like this is short-sighted.
Got it. So I got a question about herd immunity. What is herd immunity in your opinion? Where do we need to get to have herd immunity?
Yes. We have to get to very high numbers to get herd immunity. So herd immunity is basically the fact that when a virus is circulating in a population, people will get sick and they will eventually develop immunity against it. And if enough people in the community has developed immunity, then we have the virus cannot spread anymore. That's actually very effective. This is happening when we vaccinate people. We basically generate herd immunity, but we also know from our recent, experiences from people who do not want to get vaccinated, that if that level falls under a certain threshold, the herd immunity doesn't work anymore. So initially, people who don't take vaccinations will be protected because of the herd immunity, but eventually this will fall off if enough people are not participating in these efforts. So this is basically a natural vaccination that we see with a viral infection, where enough people in the community have developed immunity or antibodies or whatever the correlate is, to prevent infection and so that the virus cannot be transmitted anymore. And we know that these levels, this is very much dependent on the capability of the virus to infect other people. It's the so-called R0 factor. We think it's about 2 for this virus. And so we calculate that about 50% of the population or more, 50 to 60% of the population will have to be immune in order to make herd immunity the main driver of prevention of infection. And we are far away from this number.
Got it. So we had talked, I talked earlier this week about the importance of tracking infections, both individually, but you talked about some interesting things that are happening other places where they're tracking the virus through sewage systems. What's that about?
Yeah, I mean, I think tracking is really important. I think that's why we're developing at Gladstone a test where we connect our test directly with a mobile phone, so that the mobile phone processor, the lens is basically the diagnostic, is reading the assay, the processor is managing the whole thing, and then the GPS is helping to locate actually where the test was done. And so that's similar to what has been tried now with Facebook and other major or Google to see whether we can have a much better tracking system of people with symptoms or with positive tests. Now, one thing is that we know that the virus is not only exhaled through the respiratory, inhaled and exhaled through the respiratory tract, but it also gets into other organ systems. One of it is the gut. Many patients very early on have also complained about intestinal symptoms. And we know that the gut can become infected. And that virus is being shedded. We don't know if that is actually infectious virus. It looks as if there's no infectious virus in stool, which is good because that interrupts all the oral-fecal transmission that happens with other types of viruses. However, we detect tons of virus in the stool and in other and also in urine. And so the idea is that sewage is something that is being tracked for many indicators and quality controls. And the city of Paris has started to track the virus in there. And they could show preliminary results showing that this might be an additional means to see how high the virus is actually distributed or how prevalent the virus is in the community that the sewage is being taken from. So yes, these are unconventional methods, but I think on a population-based method, maybe something that could be useful in the future.
So you're a native of Germany. Germany is getting some good marks for how they've responded to this. I know you're probably talking to friends and family in Germany. Do you want to, can you compare the different approaches? What do you have to say about that?
Yes, I think Germany was hit very early. I think with the patient in Munich that was very early infected and actually served, I think, as a distributor in Europe for a significant line of infections. And I think the lessons we can learn from Germany is they have developed a test very early, very, very early, and they distributed it to everybody. I think our testing delay came to the point was partially due to the centralization of the testing initially in Atlanta, which was a bottleneck that we overcame very fast, but still delayed us. I think the testing is done very progressively and actively on a population basis currently, and they also have a very good medical and research system there that they have put into play, on all fronts, like we do here in the US. And I think their political system is different. I think they have, like us, states and the federal level, but I think the federal response was much more coordinated and also trusted by the population, so that all the measures and social distancing is strictly followed. So Germany currently is actually playing a little bit with fire because they will be the front riders of opening up the country. They were planning on starting limited school attendance on Monday, but actually got sued on Friday against it and had to lock it down. So there is still a lot of debate there whether they are too early, but they are confident that they have the virus under control currently, also because they have incredibly low fatality rates that really stand out around the world. But I think there's still a debate whether they're a little bit on the early side, but it will be important for us to watch.
Great. So whenever we do these sessions, I always get these great personal questions. If my daughter, who's in college, had it and recovered, how long and how safe can a mother with asthma eat together and hug her? So what do you say to folks, you know, or who have grandkids and this has gone on for a while? Do you have any general guidance on how do you proceed with loved ones?
I know it is very difficult at this moment because from the strictly scientific side, I have to tell you that we don't know what is happening and how protective the immunity will be and whether we can tell people with positive serology or a positive history that they are not going to become infected again and they will not spreading the virus to somebody else again. But I would say from a scientific standpoint, from my personal standpoint, I would say that we have a reasonable assumption that some form of immunity will be developed immediately after. I think we would have probably many more cases of repeated infection than are currently being investigated to do this. So my assumption is that the contact with the virus is inducing immunity to a certain extent, at least for the short term. The big question is really how long this will last. We know from other coronaviruses that some do induce longer lasting immunity and some don't. And so I think also back to your question from the beginning, is the severity of the infection playing a role here? Is the location where the infection has happened playing a role here? And is playing age a role also? Because we know in general that if you're older in vaccines and infections, you build less of an immune shield that is less effective. So I think there's many factors here. From the strictly scientific point, I think we don't know at the current point what is happening and how protective the immunity is. From a common sense standpoint, I think there is some immunity, but we will have to see how strong and how long it lasts.
So people are just going to have to make their own decisions based on their own health and
I would say follow the guidelines currently and try to be as responsible in the way of social distancing as you can. I know it's extremely hard with grandchildren and our loved ones, but I told my parents that they cannot have their 2 young kids currently in the house, and they're very upset with me. And I don't know if they follow it because they're in Germany, but I sure hope they do.
Wow. What do you think about keeping schools closed? I mean, there's some talk that maybe schools might be able to open later this summer with some guidelines. What do you think about that?
I thought that the school closure was a very important part of our sheltering in place, and doing it early was important. We know this from former epidemics where school closures were really the benchmark for success, basically. And I think especially with this virus, where we know that the young people often asymptomatically carry it but spread the virus, I think schools are probably a very, very difficult center of circulation, of viral transmission. And I think we have to watch this very closely, how we're going to reopen and how we're going back to a new normal here. But I think it will be, because you have to just imagine crowded classrooms or in college dorms and lunch rooms and all this. I think this is really the perfect place where the virus is going to spread.
So it turns out that one of the biggest risk factors for complications from COVID-19 is being obese. So what can you say about obesity and COVID-19?
It's interesting because my lab has a long-standing interest in actually fat metabolism and how it interacts with viruses, and we have learned quite a bit about it. That fat is actually a dependency factor, or can be supporting the virus. It's also a huge energy bowl where the virus can access energy for its own replication. And we know from Nevin's study that there is interaction of the virus or the viral proteins directly with the fat machinery in the cell. We don't know if that is the reason why this is a risk factor, but it's definitively something that we're following up on. I think the other big question that needs to be addressed here is whether, just lung health, breathing functions, are playing a role here, dependent on body weight, whether that is just a more practical explanation for this dependency.
And it also seems like men are at higher risk for death and for serious complications. Is there any explanation for that?
Well, there's lots of speculation around this because this has emerged relatively early in the pandemic already from colleagues in China. They published this beginning of the year that they see more elderly men in severe conditions than women. And that was, I think, numbers that were double or half of, the men or the women's side. And that has this held through. And I think there's, thinking about the immune system and hormones. We know that women in general might have a stronger immune system also because of their pregnancy, and the need to protect the baby. But they also, we know, have a stronger tendency to have autoimmune diseases. So the trade-off might be that you have a stronger immune system, but also maybe more propensity for the immune system to overreact. This does not seem to be the case here in this pandemic. So we don't know. A lot of people have argued that even women postmenopausal, without the necessary protection of the female hormones, are doing better than men. So they discard the hormonal explanation and squarely put it on men having more risk factors, and in general smoke more and have other more unhealthy habits. But that also doesn't seem to explain it. I'm just throwing out one interesting fact is that ACE2, the receptor, is actually located on the X chromosome, where we have, of course, 2 copies in women and 1 in men. And we just sort of starting to figure out whether there is really any, difference here that could be explaining the difference that we're seeing in the clinic.
So we're almost to the end here. I want to ask you, when you look at this virus and the work that you're doing and the work that everybody's doing, is there anything in particular that keeps you up at night that you go, oh my gosh, this, how does your mind wrap itself around what's going on in the world?
Well, I'm actually kept up at night a lot these days by many things, and I must say that the science is really that drives most of these sleepless nights, and I think it is both an extremely urgent time that we're having, but also an extremely important time as a virologist in the way that we understand this virus in a record speed, and in the way that we establish and conduct collaborations, and the way how we try to move this forward. It's really not a thinking of, oh, I do this next week. It's really, I do this in the next minute. And I think this urgency is driving everybody in the field. It's not only virologists who are working on it. I would say everybody has shifted. Every scientist who I have spoken to has shifted part of their programs and trying to contribute what they can contribute in their expertise. And I mean, the 69 or the international consortium with the 69 drugs is an incredible example of this. Most of these people are not virologists. They are experts in lung biology and pharmacology and structural biology in many different aspects. And they all have come together to just focus on this virus and their proteins and how they interact with the cell in order to develop something that is useful for the clinic in hopefully no time.
So I think you cared for patients during the HIV epidemic, right?
Yes.
Does that experience inform sort of what you do now or what you think of now?
Well, I was a neurologist in Frankfurt at the time of in the early 90s, when we really at the peak of the AIDS epidemic and no treatment. I was running an intensive care unit in neurology, and many of my patients were HIV positive and end-stage AIDS, and they all died. Nothing I could do. I had even nurses infected on the ward and dying, and it was a very, very dramatic time. I think what the pictures that I see from New York remind me very much of that time. At that time, I decided I want to become part of the solution. I want to learn more about the virus, and basically moved to New York, did a PhD in virology and HIV. And the plan was to go back to the clinic and become a physician scientist, but my experience in the lab was so profound that I got hooked to science, and I never returned to the clinic. And I would say times like this are justifying these decisions, because I think there's a lot we have to do, and a lot that keeps us up at night.
That's great. Thank you so much for taking the time to be with us today and to answer our questions. And thank you for the decisions you've made and for the work you're doing. And I also want to thank everyone who joined us for this webinar today. Our next conversation will happen this coming Monday, May 4th at noon Pacific, 3 p.m. Eastern. Dr. John Newman will be our guest. John is on the faculty here at the Buck. He's also a practicing geriatrician. He cares for patients at the San Francisco VA hospital. So here's what's cool about John. He understands both the biology and physiology of aging, and he has regular contact with older adults. He will speak to the fact that older adults and older men are at more risk for complications of COVID-19. He'll also talk about the challenges facing older adults who have survived this pandemic, and he'll give us some insights on how this pandemic might reshape the way we look at the medical care of older adults. It'll be a great session. Please pass the word to friends and colleagues. And if you're still on your computer, which I assume you are, go into session 4 and pose a question for John. A video of today's event with Melanie will be posted. We'll let you know when it's up and available. I will be back here on Monday, and I really hope you will be too. Keep the questions coming for our upcoming speakers. Enjoy the rest of your week, and please, stay safe and stay healthy. Thank you.
Thank you, Chris.
Thank you.